the pioneering invention known as the bathysphere appeared in 1920. In 1934, the 2.5-ton steel ball was lowered to a depth of 923 meters. Those inside returned with fantastic reports of creatures that glowed with their own light. People were skeptical. In 1960, a more advanced submersible carried Don Walsh and Jacques Picard 11 kilometers below the sea, into the deepest point on Earth, known as the Challenger Deep. They stayed there for 20 minutes. No one has been back since.
That remoteness, that inaccessibility has served to protect the ocean from a great many forms of human perturbation. And it means also that we seldom appreciate how significant it is in the ecology of the whole planet. And it's home to the largest animal communities on Earth, largest in terms of number of individuals, largest in terms of number of species, largest in terms of biomass. Most of the animals on this planet live in the deep sea.
Scientist Bruce Robeson has been studying deep-sea fish since the 1970s.
I had been sampling the oceanic water column with nets the way a century's worth of my predecessors had done. It occurred to me that any self-respecting desert ecologist or forest ecologist would never attempt to describe the desert or the forest without ever having been there, and yet that's what I was doing.
He hitched a ride on the Alvin, the first modern research submersible in 1974.
The dive itself was a benthic dive. We were working on the seafloor, but in order to get there, you have to pass through the water column, and I kept turning on the lights, and the pilot would say, "Turn those things off. We need them for working on the bottom," but I wanted to see what was going by my window. I was thunderstruck. There were so many more animals than I had ever imagined, particularly the gelatinous forms. They were all over the place. They were really abundant, and they were zipping around and moving and swimming and showing all kinds of active behavior. Again, something that I was unprepared for.
He still spends around 50 days a year at sea, studying the creatures that live more than 200 meters deep.
As I used to say to my students, there's only so much you can learn from a dead fish. There's a great deal more to be learned by entering the habitat, seeing the animals in their natural environment.
The North Pacific coast, just south of San Francisco, is an ideal place to start any deep water expedition. The ocean floor plunges down from the shoreline. Marine life also flourishes along the coast that's part of a protected reserve. It seems that every trip leads to another new discovery. Compared to the early iron spheres and submersibles first used to probe the deep, modern-day explorers have state-of-the-art diving and imaging technology at their fingertips. On the way down, the submersible passes through the upper waters, home to familiar species like sardines, sharks, and everything between. Sunlight and oxygen support life in this upper zone. At a depth of around 200 meters, the mesopelagic, or mid-water zone, begins. If any sunlight does exist, there's not enough to see or support plant life. Without any light from the sun, creatures down here make their own.
Bioluminescence is apparent in animals throughout the water column, but as you go deeper, bioluminescence becomes more and more important as the only means of communication that we're aware of.
The few creatures that can't produce their own bioluminescence harness bacteria that do. These spectral animals don't glow in the dark to see where they're going, but use light to communicate. Flashes of light are used to emit a welcome or a warning to stay away. Certain species of shrimp can even vomit a bioluminescent irritant that temporarily incapacitates an attacker.
My colleague Edie Witter says that, looked at objectively, bioluminescence is the most important communication form on Earth. And I'm sure she's right, because of the vast numbers of plants and animals that create light in order to communicate with one another, to avoid predation, to attract prey, and for all kinds of other interactions.
In other words, more creatures use light to communicate than all forms of sound put together, including the vast array of human languages. Bioluminescence is just one of many ingenious adaptations animals have evolved to survive and thrive in the deep sea. Some 600 meters down, the capture of specimens like this vampire squid helps researchers make sense of a world that's still relatively unexplored. But other deep-sea creatures are not so easy to capture or explain.
Definitely.
Possibly the longest animal organisms on Earth, siphonophores are carnivorous predators, but so delicate, they disintegrate when netted or grasped.
The largest siphonophore we've ever measured was 41 meters in length. Siphonophores are gelatinous predators that send out a curtain of tentacles that will capture anything that happens to blunder into it. They are unusual in that they're colonial animals.
It's a colony that behaves like a single organism. They're made up of multicellular individuals that work together, although each has a specialized function. A siphonophore is like a cooperative made up of thousands of conjoined twins, each designed to perform a specific task.
Like ant colonies, there are specialized individuals who have particular functional roles within the community. There are propulsive units. There are individuals that are reproductive elements. There are individuals whose role is to catch and digest food.
Nothing like it can be found on land. Experts can't decide how to classify it.
Do you treat it as a colony? Do you treat it as an individual? Or do you think of it as some sort of superorganism, which is often the fallback position? Well, it's special, it's not like anything else. We'll call it a superorganism and leave it at that.
Other creatures that live alongside these superorganisms have also evolved superadaptations to protect themselves from lethal stings and to feed off those that can't.
Macropin is a little fish that we have known about for a great many years from net-caught specimens. When we first came across one, we were astonished to see that there was a transparent shield over the top of its head. Every specimen that I'd seen was drawn without transparent shield. We were well aware that they had tubular eyes that looked upward. And it always puzzled me that they seemed not to be able to include their mouth in the field of view of their upward-looking eyes. And how can you feed effectively if you can't see what you're eating? Well, as soon as we began to observe these animals in their natural habitat, everything came clear. And we've been able to determine that the eyes are capable of rotating from their upward-looking position to a forward-looking position. And these animals probably swim along beneath large siphonophores. When it sees something in the tentacles, it rotates the body and the mouth upwards, grabs the food, rotates the body back down, and proceeds down the linear smorgasbord of food that the siphonophore provides. The transparent shield protects them from the stinging cells in the tentacles of the siphonophore that they're swiping food from. The paradox of how that fish could see to eat was suddenly resolved by the fact that we could enter its habitat and watch it firsthand. We have been able to bring 2 specimens of Macropinna alive to the surface to confirm the observations that we'd made with the ROV. It's important to be able to make measurements in the lab that confirm what you see in the ocean, and vice versa. If you do laboratory experiments, it's very important to be able to confirm your results in the natural habitat.
But not today.
It seems the Macropinna is also far too evolved to fall for a human trap. Deep-sea expeditions have revealed a magnificent array of animal adaptations, and they've probably only scratched the surface.
We don't know what we don't know, that's for sure. We suspect that there's a hell of a lot we don't know, and we can categorize it, but there are only a few places where we could say we have even gotten a good start.
Until recently, deep-sea fish were out of reach of commercial fishermen. Fish in shallow and coastal waters were easier and cheaper to harvest. But when these fish became scarce, trawlers looking to make up the shortfall steered a course for deeper waters. With little idea or regard as to what damage might be done, nets were dropped and dragged up to 1 kilometer below the waves. On the seamounts off New Zealand, deep-sea fishermen struck gold, or orange, to be exact.
Science had known about orange roughies since the late 1800s, but basically they lived undisturbed for many millennia until the mid-1970s when they discovered orange roughy in New Zealand waters on the East Chatham Rise.
The orange roughy was an excellent commercial catch. Its mild white meat stayed firm when thawed after freezing, and there were no restrictions or quotas. When other concentrations of orange roughy were found in the Pacific, Indian and North Atlantic, fishing boats plundered the orange bounty. So many were caught